Eric Rubin here, and as Monica indicated, I am a co-moderator for this session. I would like to thank the speakers for the excellent presentations. We will now begin a panel discussion, and I would like to remind public participants that we encourage submission of questions and comments on the public chat, which is located below the webcast video. So I'm going to lead off with the first question, and this is for Efrat. You mentioned that the GIANT study includes dose modifications for the 2 chemotherapy regimens that are being tested. Can you explain what these are and how the doses were chosen? Also, can you also comment on the target sample size and how accrual is going, as well as what is the overall survival effect size that the study is powered for, and how does this compare to the targeted OS effect size for NAPOLI-3?
Wow, a lot of really good questions. I'll start with the dose. So the dose was clearly a huge discussion and back and forth between all the people involved in the study development. I would say all the oncologists agreed that the gemcitabine and Abraxane arm was what we all use as standard regimen for this patient population. And despite the lack of actual prospective data here, this was evaluated through a huge questionnaire from the Pancreatic Task Force and cooperative group members. And everyone agreed that dose reduced every other week gemcitabine and Abraxane is the regimen of choice for oncologists. So that was the decision for that arm. The 5-FU Onivide, the reason we selected to use a dose reduction was based on data provided from the second-line setting by the company, showing that patient who received 50 milligram rather than 70 milligram actually did not have a significant difference in outcome. And some of them were able to tolerate the drug for a longer period of time. And since we are targeting a vulnerable patient population, it seemed appropriate to use that dose as a start, making sure that we don't expose these patients to too many toxicities. So that's your first question. Your second question was with regards to overall survival, if I remember correctly. So again, this was a big discussion. And I want to point out the NAPOLI-3 study targets a very different patient population. So I think the comparison in terms of the overall survival endpoint on that trial versus our study is a little bit flawed because the patient population is very different. And especially when you're thinking about an overall survival endpoint where majority of patients will end up from the NAPOLI-3 study, end up getting second-line therapy, and that will definitely affect that outcome. I don't know how many patients from our study will ultimately receive second-line therapy. That's hard to predict, given, again, we're targeting a vulnerable patient population. And I can remember all the statistics off the top of my head, but we were looking for about a 3-month improvement in outcome on this trial, thinking that the 5-FU Onivide group will do better than the gemcitabine and Abraxane. And as you all know, survival in metastatic pancreatic cancer is quite dismal. But I believe the rationale came from the data that we were able to collect from studies done on gemcitabine and Abraxane dose-reduced therapy in various populations that are not fit patients, and trying to extrapolate from that and from second-line use of 5-FU Onivide on what type of benefit we would be able to see.
Thank you.
Wow. Okay, well, so many questions here and so little time, right? So Eric, I'm going to move forward with one of your questions then next and ask this one of Kevin. Kevin, can you comment on accrual rates for the TRIO cohort 2 versus 3? Were TRIO sites selected based on a likelihood of being able to enroll older patients?
Thank you for your questions. Can you hear me, Dr. Patrinoli?
Yep.
Okay. So we were actually quite pleased. And, I will say, if there's one thing I've learned in this business of developing drugs as a sponsor it's to listen to your investigators. And the whole idea really was inspired by a number of our investigators coming to us and saying, hey, this drug could be quite good in the elderly population, given the low neuropathy rate and the fact that you can take it at home and so on. And so there maybe was some self-selection there. The studies enrolled well. All of our studies, including the 69 patient elderly only study, enrolled well. I think that study enrolled in about 1 year. And I would say we didn't specifically select centers that targeted just the older population, but we did include, as I mentioned, community oncology sites in all of our studies, which in hindsight, I would say, I don't know if we really expected this, but now it becomes more clear, often have a broader patient population than patients who seek out, major teaching hospitals in major cities. And so we were quite pleased with that. I would say we had a broad base of centers, and that helped us more than it was specifically targeted towards an older population.
Thanks. I'll move on with a question for Dr. Stone. These relate to the CPX-351 drug in phase 3 study. This is an interesting drug. I believe it's the first and maybe the only example of a fixed drug chemotherapy combination in oncology, two fixed chemotherapy drugs given in a liposome. I noticed that the phase 3 selected an age group from 60 to 75. I'm curious as to what was the rationale for the selection of this specific age group, if you could comment on that. And also, was the initial dose finding with this drug done with this particular age group in mind?
Those are great questions, Eric. The answer to the first question is basically the drug company cherry-picked from a phase 3 trial of 3 and 7 versus this drug in a bunch of in older adults, which was a negative trial. But they noticed the subgroup, unplanned subgroup analysis of those who had secondary leukemia. So they found the water was not too hot, not too cold. You wanted patients that were relatively resistant to 3 and 7, where something that might be a little bit better was available. So they bit the bullet, did a large phase 3 trial, in not only just older patients, but older patients who had documented secondary leukemia. They had to have a prior bone marrow stem cell disorder. And the dose finding was done in patients of all ages. And so really what is done in older adults with AML is they're divided into so-called fit and unfit. So the fit are presumed to be able to tolerate full doses of your typical drugs. So the phase 1 trial looked at patients of all ages, came to a dose that could be given to patients of all ages, and it was just slotted into older adults. So once you're fit, you can get whatever a young person gets, more or less.
Thanks, Rich.
Okay, great. Thanks, Rich. Okay, let's see. I've got one here for Monica Giovannini. Can you comment on geometry cohort enrollment rates relative to other Novartis lung cancer studies that enrolled a much less restricted population? And once the correlation between MET exon 14 mutations and older age was known, did you consider specifically adding sites with expertise in enrolling older patients, or did it just flow from your original site team?
So thank you so much. Thank you so much for the question. So as I explained during the presentation, when we started the study, there was really no knowledge about MET exon 14 skipping mutation. So we started enrolling patients with MET amplification, with different level of amplification. And the enrollment rate was, let's say, what expected for general non-small cell lung cancer enrollment rate. So we're having in the very low gene copy number cohort that resemble much more the unselected non-small cell lung cancer patients. So because the MET amplification can exist as not a molecular driver across non-small cell lung cancer patients, the enrollment rate was very high. We had 15, 30 patients per month, very, very easy. Then going higher in the gene copy number, this became a more selective patient population. Shifting to MET exon 14 that we included later on, the enrollment rate was very difficult, especially at the beginning, because in the scientific community, there was really not much knowledge about METex14 as a target. So we're about 1 patient per month for several months. Then as soon as the scientific community got a bit more aware of METex14 as a target, and we were able to show our initial data together with our other MET inhibitor in the same field. Then this awareness prompted the physician to test more extensively these patients, and we have been able to but also learn that we don't need to pre-select this patient based on gender, as it happens for other molecularly selected non-small cell lung cancer groups, not based on smoking histories, not based on histology, and certainly not precluding the screening for molecular pre-screening to more elderly patients that eventually became the target of this cohort. So it was a very nice learning, and physicians, they went through all this massive pre-screening effort, because we say it's 3-4% of the patient, but we had physicians who screened 100 patients, they had 0 patient positive. So is of course not it doesn't depend on the pool. But all this learning prompted this physician to test, and we ended up enrolling in the moments when we had the peak of enrollment after showing the activity in second, third line and the initial data in first line. We end up with 5 patients per month. So it's a challenge patient population, but for those of us who work in molecularly selected patients, 5 patients per month in a 3-4% patient population study is not bad, with the number of sites that I was mentioning is about 130 sites globally.
Thanks. I have a question for Hans. So for the interesting 75111 study, there were impressive efficacy results with progression-free survival with the addition of metronomic cyclophosphamide, oral cyclophosphamide. I'm curious if the addition of this agent increased toxicity, or was it similar to the other arm?
So there was nearly no difference in toxicity, only slightly more lymphopenia. And we have just submitted the manuscript on quality of life that was identical in the 2 arms.
Thank you.
Wow, that's great. So maybe now we could go back to Efrat. Efrat, your study was, it's wonderful to see all these different designs, right? This is a fantastic roadmap for others. We've really got the spectrum here. Your study was specifically designed for older and more frail patients. And I'm curious as to what the reception of this study was among the trial participants and the treating clinicians. Did you have any accrual problems, or was it really embraced from the beginning that you were going to be studying a pretty vulnerable group?
So the study is ongoing. I can say that through the cooperative groups, it's open through all the cooperative groups and the NCORP site and community sites underneath the cooperative groups in over 300 sites, I believe, in the country that are open for accrual. It's been activated for a little bit less than 6 months, and we, as of yesterday, have 18 patients enrolled. So I think within the timeline, this is kind of a pretty good timeline. I can tell you that the reception was really incredible. I think all the oncologists were excited to finally have a study that fits the patients that they see in clinic. And I've been getting emails from people basically surprised as to how easy it is to enroll the patient, because the eligibility is open, and sometimes even asking me question, how come the patient can go on it if they have, breast cancer 2 years ago? But really, a small breast cancer in an older woman that was resected is the least of her issues when she has metastatic pancreatic cancer. So I think physicians are really excited to have a study that is applicable to the people they see in clinic, that allows them for easy access to a trial that will provide input for for the care of these patients. And I was asked earlier about the overall survival and the endpoint. I keep saying that in this trial, and I think that's true for a lot of the elderly specific trials as we try to develop them, whatever answer we get is a good answer because we really don't know what to do with these patients. So if the trial is a positive or a negative study, it doesn't matter. It really tells us how we need to approach the care of these patients. And we gain so much knowledge from it. So yeah, I just hope this is the first in many of these kind of trials.
Thanks. I have a question. I think it's from Monica for Kevin. Congratulations on your successful trials with registrational intent to develop a therapeutic agent with features suggesting that it's particularly suited for older patients. Do you plan to move this agent into testing in the adjuvant setting? And if so, will your approach to engaging older patients change?
Great two-part question, Dr. Rubin. I think the answer to the first part is yes. Hopefully post-approval, knock on wood, in the metastatic setting, we would hope to move earlier in treatment to the early breast cancer setting. And that is in fact one of the appeals and one of the many trial designs we're evaluating now. Of course, the good news is there are more and more options today in the adjuvant and neoadjuvant setting. But just like in the metastatic setting, where maybe sometimes hitting the patient with the hardest therapy may not be the right answer for all patient populations, maybe there is a patient population like the older adult population in the adjuvant or neoadjuvant setting where Tessa-Taxol may be a good alternative to some of the more aggressive regimens. So I would say we are very hopeful that we can move earlier in treatment, and we are very much evaluating with our partners at investigator sites different options for how to do that. Great.
And
well, go ahead.
So regarding targeting MET-exon 14 skipping in the adjuvant or any earlier line, like in the adjuvant setting, first of all, we know that the MET-exon 14 is a molecular driver, and it doesn't seem to, the prevalence doesn't seem to change depending on the stage, even though there are groups that demonstrated it seems to be a bit more frequent in the earlier stage, even though it has not been fully demonstrated in larger data set. There are, of course, intention to move in earlier settings. However, the pool of patient becomes smaller in non-small cell lung cancer in the adjuvant, in neoadjuvant, and this brings even bigger challenges in addressing these in prospective studies, because of course we know what's the proportion of the advanced versus the earlier stage in this type of tumor, which is very different from breast cancer, for example. So these increase the feasibility challenges, but of course, seeing the activity of other targeted therapy in adjuvant setting in non-small cell lung cancer, this prompted us to think about it. So certainly something we are working on it, and we will probably not work necessarily to go into institutions that have a target for geriatric oncology, not necessarily, because as I was explaining in the presentation, these physicians are used to treat elderly patients just by default because of the age of diagnosis of non-small cell lung cancer. MET-exon 14 are older than the usual, but not that older that require these geriatric preselection.
Great. So, Hans, you started to address the issue of endpoints, and composite endpoints. And oh my goodness, I think that aspect of your talk could take an entire workshop just itself to think about how we would do that. We have a question for you from the chat. Dr. Mohile wants to know, how would you propose to measure tolerability in trials for older adults? Historically we use CTCAE by clinicians, but are there other endpoints for tolerability that you could recommend that should be routinely measured?
Well, I think that some quality of life measurements or maintenance of functionality is extremely important as co-primary endpoint or as part of a composite. And And but I also think that the ideal endpoint depends a lot on the setting. In an adjuvant setting, you will need a very different endpoint than in a setting where you have treatment with a lot of toxicity. But I want to emphasize the importance of integrating geriatric assessment, because I have the impression still that in mainly pharma-sponsored trials, we don't measure geriatric assessment, and this is a pity. It does not allow us to evaluate the tolerability in relation to frailty.
Yeah, good point. Eric, you want to keep it going here? I think we can give it one or two more here, just if you like.
So I noticed there's a comment from Dr. Kleppen, and maybe it sounds like there's an AML reference to this, so maybe I'll ask Dr. Stone to comment on this. Dr. Kleppen notes that it's important to highlight that some therapies may work better in older adults, which is not assumed, particularly when therapeutics harness the principles of geroscience. So maybe, Rich, do you want to comment on it, or maybe Heidi as well, if you'd like to add to that?
Well, I'll just say briefly that there may be therapies that are better based on the biology for older adults, like the aza-ven I talked about, inciting venetoclax. That was for patients greater than age 75. And maybe that's because they have a type of tumors that will be more likely to respond to low-dose therapy compared to high-dose therapy. But I'll leave it there and let Heidi comment further.
Oh, thanks. Yeah, I just was picking up on that thread from the CPX conversation that, the biologic changes of aging that affect both the tumor and the patient can make older adults in some cases more likely to benefit from certain therapies if we're targeting the therapies towards those biologic mechanisms specifically. And I think we're not going to pick up on some of those observations where that might be the case if we're not studying older patients in the first place, particularly on the early phase trials.
Absolutely. And I think it's important to ageism is probably, as you well know, I mean, it's not about your chronological age. It's, as we've all said, it's about your fitness and about your biology. It is that possible for an older adult to have a younger person's leukemia and vice versa.
Okay, fantastic. Well, let's draw this one to a close. We're going to take a break. If we could reconvene at 2:45 for the second half of our session. I'll just remind everyone that the second half of this session is going to be about policy opportunities with a focus on registrational intent studies. So we look forward to seeing you all back in about 13 minutes at 2:45. Thanks.